Today's discussion centers on a fascinating chapter from Sherris and Ryan's Medical Microbiology, focusing on the basic concepts of infection. It's a field constantly evolving, isn't it?
Absolutely. The landscape of infectious disease is dynamic, shaped by factors ranging from antibiotic resistance to emerging pathogens and even the impact of global events.
Precisely. Sir William Osler's quote, "Humanity has but three great enemies, fever, famine, and war. Of these, by far the greatest, by far the most terrible, is fever," rings true even today, though the specifics have shifted.
His words highlight the historical dominance of infectious disease, a dominance that has lessened in many parts of the world, but remains a significant threat globally.
And that brings us to the core of this chapter. How has our understanding of infection evolved, and what are the key challenges we face today? Let's start with the historical context. The chapter traces the advancements from the pioneering work of Pasteur and Koch to the modern era of molecular biology and genomics. What are some key milestones that shaped our understanding?
The work of Pasteur and Koch established the germ theory of disease, revolutionizing our approach to understanding and treating infections. This led to the first golden age of microbiology, where many bacterial diseases and their causative agents were identified. The development of vaccines and antimicrobial agents in the 20th century dramatically reduced mortality rates in many developed nations.
But that chapter also emphasizes that this progress hasn't been universal. Infectious diseases remain a leading cause of death in many parts of the world.
Indeed, access to sanitation, vaccines, and antimicrobial therapies is unevenly distributed globally. This disparity contributes to the persistent burden of infectious diseases in less developed regions.
The chapter then delves into the microbial world itself, categorizing infectious agents by increasing complexity: viruses, bacteria, fungi, and parasites. Can you elaborate on the distinctions between these groups?
Viruses are the simplest, lacking the cellular machinery for independent replication. Bacteria are prokaryotic cells, lacking membrane-bound organelles. Fungi are eukaryotic, with cell walls composed of unique polymers. Parasites encompass a vast range of organisms, from unicellular protozoa to multicellular worms, all sharing a eukaryotic cell plan. Each group presents unique challenges in terms of diagnosis and treatment.
A crucial concept highlighted is the human microbiota, the vast community of microorganisms residing on and within us. What's the significance of this microbiota in health and disease?
The microbiota plays a multifaceted role. It contributes to our immune system development, aids in nutrient metabolism, and can even protect against pathogenic organisms through competitive exclusion. However, disruptions to the microbiota, such as through antibiotic use, can create opportunities for opportunistic infections. The balance is delicate.
The chapter mentions the carrier state, where potentially pathogenic organisms reside without causing disease. How do we understand this phenomenon?
The carrier state highlights the complex interplay between host and pathogen. Factors like host immunity, pathogen virulence, and environmental conditions influence whether carriage leads to disease. It's a dynamic equilibrium that can be easily disrupted.
The discussion then shifts to the mechanisms of infectious disease, covering epidemiology, pathogenesis, and immunity. Let's start with epidemiology. How does understanding the spread of infectious agents inform our prevention strategies?
Epidemiology is crucial for understanding disease transmission. Identifying modes of spread airborne, foodborne, vector-borne, or direct contact allows us to implement targeted interventions, such as sanitation improvements, vaccination campaigns, or vector control measures. Modern society, with its interconnectedness, presents both opportunities and challenges for disease spread.
And pathogenesis. What are the key steps involved in a pathogen causing disease?
Pathogenesis involves multiple steps: initial attachment to host cells, invasion of tissues, evasion of the immune response, and damage to host cells or tissues. Virulence factors, specific traits that enhance a pathogen's ability to cause disease, play a critical role in each of these steps. Understanding these mechanisms is essential for developing effective therapies.
Finally, immunity. How does the immune system respond to infection, and how can this knowledge be leveraged for prevention and treatment?
The immune system employs both innate and adaptive mechanisms to combat infection. Innate immunity provides immediate, non-specific defense, while adaptive immunity develops a targeted response to specific pathogens. Vaccines exploit this adaptive response, stimulating the production of protective antibodies or immune cells. Understanding the immune response is crucial for vaccine development and for designing therapies that enhance the host's immune defenses.
The chapter concludes by discussing the clinical aspects of infectious disease, including diagnosis, treatment, and prevention. How have diagnostic methods evolved, and what are the current challenges?
Diagnostic methods have advanced significantly, from traditional culture techniques to molecular diagnostics that detect specific genetic markers of pathogens. Rapid diagnostic tests are crucial for timely intervention, particularly in outbreaks. However, challenges remain, including the emergence of novel pathogens and the need for more sensitive and specific diagnostic tools.
And treatment. How effective are current antimicrobial therapies, and what are the concerns regarding antibiotic resistance?
Antimicrobial therapies have revolutionized the treatment of infectious diseases, but the widespread use of these agents has driven the selection and spread of antibiotic-resistant strains. This resistance poses a major threat to global health, necessitating the development of new antimicrobial agents and strategies to combat resistance.
What about prevention? What are the most effective strategies for preventing infectious diseases?
Prevention relies on a multi-pronged approach, including public health measures like sanitation and hygiene, vaccination campaigns, and the development of new preventive strategies. The chapter highlights the importance of both live attenuated and inactivated vaccines, as well as newer approaches like mRNA vaccines. Each strategy has its own advantages and limitations.
Let's revisit the microbiota. The chapter emphasizes its role not only in preventing infection, but also in contributing to certain diseases. Can you elaborate?
The microbiota's role is complex. While it generally protects against pathogens, dysbiosis or an imbalance in the microbiota can contribute to various conditions, including inflammatory bowel disease, obesity, and even certain autoimmune disorders. Research into the microbiota's role in health and disease is a rapidly expanding field.
The chapter mentions probiotics and fecal microbiota transplantation as ways to manipulate the microbiota for therapeutic benefit. What's the current status of these approaches?
Probiotics, while showing some promise in specific situations, have had limited success in broadly altering the microbiota. Fecal microbiota transplantation, on the other hand, has shown remarkable efficacy in treating recurrent Clostridium difficile infection, highlighting the potential of restoring a healthy microbiota. However, more research is needed to optimize these approaches and to understand their long-term effects.
The chapter also touches upon the emergence of new infectious diseases and the threat of bioterrorism. What are some of the key challenges posed by these threats?
Emerging infectious diseases like HIV and COVID-19 highlight the unpredictable nature of infectious disease. These pathogens can rapidly spread globally, causing significant morbidity and mortality. Bioterrorism poses an even more sinister threat, with the potential for deliberate release of highly virulent pathogens. Preparedness and rapid response are crucial for mitigating these threats.
How can we better prepare for these emerging threats?
Enhanced surveillance systems, rapid diagnostic capabilities, effective vaccines and therapies, and robust public health infrastructure are essential for responding to emerging infectious diseases and bioterrorism threats. International collaboration is also crucial for sharing information and coordinating responses.
Looking ahead, what are some of the most promising areas of research in infectious disease?
Several areas hold great promise. Research into the microbiota's role in health and disease, the development of new antimicrobial agents and strategies to combat resistance, the development of novel vaccines, and the application of advanced technologies like genomics and proteomics to understand pathogen evolution and virulence are all crucial areas of focus.
The chapter emphasizes the importance of understanding the molecular mechanisms of pathogenesis. How is this knowledge being applied?
Understanding the molecular mechanisms of pathogenesis allows us to identify specific targets for new therapies and vaccines. This knowledge is crucial for developing more effective and targeted interventions. Genomics and proteomics are playing an increasingly important role in this area.
The chapter underscores the importance of interdisciplinary collaboration in addressing the challenges of infectious disease. Why is this collaboration so crucial?
Infectious disease is a complex problem requiring expertise from various fields, including microbiology, immunology, epidemiology, clinical medicine, public health, and even social sciences. Interdisciplinary collaboration is essential for integrating knowledge, developing effective strategies, and translating research findings into practice.
How can we foster this collaboration more effectively?
Increased funding for interdisciplinary research, the creation of collaborative research centers, and the development of training programs that emphasize interdisciplinary approaches are all crucial steps. Promoting communication and knowledge sharing between different disciplines is also essential.
The chapter concludes with a powerful message. The battle against infectious diseases is ongoing. What are the key takeaways for students of medicine?
Students of medicine need a strong foundation in microbiology, immunology, and epidemiology to understand the complexities of infectious diseases. They must also be aware of the challenges posed by antibiotic resistance, emerging pathogens, and bioterrorism. A commitment to lifelong learning and interdisciplinary collaboration is essential for effectively addressing these challenges.
The chapter highlights the need for a holistic approach, considering not only the pathogen, but also the host, the environment, and the social determinants of health.
Absolutely. A holistic approach is crucial for understanding and addressing the complex interplay of factors that contribute to infectious diseases. This includes considering social, economic, and environmental factors that influence disease transmission and outcomes.
The role of public health is paramount in preventing and controlling infectious diseases. How can public health initiatives be strengthened?
Strengthening public health infrastructure, investing in surveillance systems, promoting vaccination campaigns, and implementing effective infection control measures are all crucial steps. Public health education is also essential for empowering individuals to protect themselves and their communities.
And the importance of continued research and innovation cannot be overstated. That was a great discussion. Thank you for sharing your expertise and insights on this crucial topic.